Hello everyone, I am Hakime. I am a postdoctoral researcher at Stegle Lab in Heidelberg, German Cancer Research Center, and I am very pleased to be here and sharing our ongoing work with you. So it's about using variant impact-based models for cancer subtype analysis, but rather than the patient similarity networks, the focus of this talk would be more on how we can derive these embeddings that we can use to actually compute these patient similarities. So as we already see today that there have been so many works on identifying cancer subtypes, and most of the time because the identification of these subtypes can help the grouping of patients with similar clinical phenotypes, which then in the downstream can help enhancing the treatment strategies. And that is why so many researchers have been trying to develop methods, most of the time including either clinical and or multi-omics data with different integration strategies such as similarity network fusions or machine or deep learning methodologies. However, the orthogonal feature of these somatic mutations that are actually could be the drivers of some of the molecular changes in these omics types, such as expression, have not been fully explored yet. And the focus of this talk would be how can we use these existing somatic mutations, in particular single nucleotide variations, in the identification of cancer subtypes, in particularly trying to obtain a patient level representation. So among the existing studies that are trying to derive patient-level representations from the somatic mutations are one set of very prominent works, namely referred as mutational signatures. And in this type of studies, we mainly assign the mutation into one of these 6 mutation types, and then we take a look at the 5' and 3' nucleotide contexts, and then we basically obtain a frequency vector of 96 dimension. And the most favorite method of the researchers is the matrix factorization to get to a latent and low dimensional embedding. And another study I want to mention is from a few years back, Zhang and co-authors, where they were using mutation features that are also extracted from sequences, but slightly increasing the sequence context compared to the mutational signatures, going up to 6 nucleotides, but also adding additional features like which genomic bin these mutation falls to. And then they basically linked these mutation embeddings into the patients by using their IDs. I referred as the mood space. This work aims to pull the embeddings of the mutations together in the embedding space because they belong to the same sample. And by doing that, they're also expecting the patients that are having similar mutation profiles also coming closer to each other in the embedding space. And this would imply that maybe these samples actually belong to the same subtype or share some kind of similar characteristics. However, with both of these studies, the information or the embedding that is extracted for the mutation only considers a very limited sequence context, which is most of the time, let's say, up to maybe 15. But if you were in the regulatory systems course yesterday, you would have probably seen some of the presentations that even an enhancer that is megabases away might really have an impact on the regulatory system. So including larger sequence contexts are really crucial, especially when we are dealing with these somatic mutations. Luckily, there is already been a lot of work, also again mentioned a lot during the conference, that the deep learning based methods that are based on actually really large sequence contexts, and as in given in this example here from last year by Chen and co-workers from the Trojanski lab, where they basically trained a convolutional neural network based models on 4 kilobases of genomic sequences, and where they were predicting over 21,000 chromatin profiles, including transcription factor activity or DHS activity. And Also, one cool thing they also did was, since they already had a pre-trained model, they tiled the whole genome in overlapping windows, and they extracted 30 million sequences. And then afterwards, they used this pre-trained model to obtain this chromatin profile predictions, and then they performed clustering. And then in the end, they arrived 40 different sequence classes that are actually representing some of these important transcription factor activities, such as the CTCF or some basically promoter-related activities. So in this work, we wanted to make use of this functionally enriched annotation, which is actually 40-dimensional, but gives us an information about what kind of regulatory activities these mutations have an impact on. And a very quick summary of how actually you can compute the impact of a mutation from such pre-trained model is basically you have the mutation letter, and then you extend the sequence context for the requirement of the algorithm. And in this case, for say, it was 4 KB. And then you simply feed the wild type sequence, and then you feed the mutated sequence with the single mutated letter, and then you obtain the reference and alternative prediction, and the difference between them gives you the impact of the mutation, which is actually interpretable, that you can see what kind of regulatory activities it actually has an impact on. So coming to our model, which we are currently prematurely named as DeepMut-Ag, it tries to learn an aggregation model, and we have the 3 main motivations. The first one is actually, as I just mentioned, utilizing these enriched mutations that are actually available in the literature as in the forms of pre-trained models. And the second, to have an aggregation of these mutation annotations on a patient level embedding. And the third is to inform this aggregation model from an existing modality. So this could be either expression, methylation, or clinical data, but this other modality already encodes the structure that is potentially there for the disease. So the way that we are learning this aggregation model is inspired from a research area called metric learning. And the common application examples of metric learning is when you have an image and then you have a text, and this text is actually the caption of the image that is describing what is happening in the image. And then in the ideal case, you want to pull the embedding of the image and the text together in the space because they represent the same entity. So we borrowed this idea, and we have these patient-level mutation aggregation embedding, and then we have another type of modality in this example, which is expression, and we basically want to pull these embeddings together in the embedding space because they belong to the same patient. Of course, in order to arrive this mutation-based patient embedding, we have to have some kind of aggregation model, and this is actually the model that we are focusing on to learn, the aggregation model itself, and it actually needs something as an input, which is the embeddings that are coming from the same model I just recently introduced, which has 40 dimensions that actually gives us information about the regulatory impact of the mutation itself. A quick look into what is actually the aggregation model. It is based on a set neural network, which is permutation invariant, and it is quite simple. It has one linear layer and a non-linearity, and then we have this pooling function, which could be sum, max, or it also could be a self-attention mechanism, and then followed by another linear and non-linearity. And our input is basically a 3D tensor. We have the samples, mutations, and annotations, and the output is basically an embedding for the sample. And I want to quickly mention that we just had a paper in our lab with also collaboration in Gagner lab in Munich that was actually using this set networks, which we just put into bioRxiv 2 weeks ago, where we basically use this set network architecture for rare variant association studies. So coming to how we can train this model is we are actually using a self-supervision, and this means that we need to assign some soft labels. And in this case, so we have these binary classes, and one of the classes is actually the correct pairing, which means that for each sample we have the correct modality, one coming from the mutation aggregation and the other coming from, again in this example, expression. And also in another case we have these negative samples, again soft labels, which are actually constructed by the mismatch pairs. And the decision of how we select the mismatch pairs is really an area of exploration, but right now we only use the constraint that the mismatch pairs doesn't share any mutation. For the training, we are using the contrastive loss, and this implies that if we have a sample and its correct modalities, mutation embedding, the expression embedding, then we expect that in the embedding space that they would be close to each other while the distance is being close to zero, which forces the loss to become zero. For the negative pair, we have a margin, a hyperparameter M, which refers to a margin. So it could be any value, but usually like between 0.5 or 2. And the goal is to pull the mismatch pair, the distance between the mismatch pair larger than the margin. So as a proof of concept, we applied this methodology on TCGA breast cancer cohort using the samples from whole exome sequencing, and we had 985 samples, and we computed these say-based embeddings, and we only considered the mutations that has these impact scores over 0.01. And as you can see from the left side, that usually the average number of mutations belonging to one patient is between 100 and 200. And on the right side, you see the plot of the mutations coming from the say embeddings. So each dot here is a mutation. And we can see that most of the time, the mutation in this cohort, the breast cancer cohort, is enriched for the CTCF enhancer or transcription activity. So the variation is only major for these three regulatory features. For the training, we created a training. We separated the samples into train and test sets. For the train samples, now these become our true pairs, and then we generated mismatch pairs by the triple of size. Again, this is another choice that needs to be fine-tuned. And here are our first proof of concept results. This is mostly the results I will be showing you now on the PCA-based visualization. And it is for the validation sample that were set aside and wasn't a part of the training. And here, the two plots that you are seeing are our baselines, which are basically simple aggregation of these three-dimensional tensor that I mentioned, mutations, samples, and the annotations that are basically compressed across the mutational axis. So either by using the sum or max, and each dot here represents the patient, and we colored them by the subtypes that are coming from the already established work from the literature. And as you can see, there is not really quite a separation. And this is now coming from our still growing method. And it seems slightly better than what is out there in the baseline. And we can have see some small separation, at least in one of the subtypes, but there is still a lot of work to do. And as I mentioned during the steps, that there are so many design choices that actually derives how the aggregation network itself is trained. So in summary, I want to emphasize that while dealing with the somatic mutations, and then as I mentioned, I was mostly referring to single nucleotide variations using already available models, pre-trained models that are trained on the large sequence contexts, and that gives us information about the regulatory impact is quite powerful, and I think there are a lot to explore on that part. And then we proposed a deep learning method that was actually quite shallow. But then we tried to guide this aggregation model through using another data modality. And actually, the main message of this studies using this pre-trained aggregation model afterwards to generate patient-level embeddings. And as in our case, we have a specific leiomyosarcoma cohort in our collaborators in the National Center of Tumor Board, and it is not quite really established what are the subtypes, and having these pre-trained model in the near future, hopefully more fine-tuned, will help us to identify the potential patient similarities in that cohort as well. So with that, I am thanking Oliver and all my lab members for the helpful discussions on the methodology, and also our clinical collaborators, also our next door neighbors for their help on discussions on the data. And thanks a lot, merci beaucoup.